the whole enzyme, you can say. First of all, let's say it's the whole kulu, like big enzyme. Okay, so it's made up of smaller parts, right? One part of hollow enzyme is an apoenzyme, which I said that it is a protein part of it. But then this hollow enzyme is also made up of something called as a non-protein part. Non-protein part. Okay. So now the point which you have to be very, very careful, where the students do get confused. What is the difference between a cofactor and a coenzyme? Can anyone tell me what is the difference between cofactor and coenzyme? Do you know what is the difference? Nobody? Okay. This non-protein part is of two types. This non-protein part is of two types. Let me see. For metal, we have cofactor for metals. Very good. Yes, Rand, please go ahead, dear.
Cofactor is necessary for catalytic function,
Okay.
and the coenzyme acts as a cofactor.
Okay. See, does that mean that coenzyme is not required for its catalytic action? You absolutely said it correct, right? But I just wanted to clarify your doubt. Does that mean only cofactor is required for the catalytic action of enzyme? We don't need coenzyme?
No, we need both.
We need both, right? So coenzyme for the small, absolutely. See, this is the difference. Your answer is correct, okay? But I just wanted to clarify one thing, that this non-protein part can be a cofactor. It can be a cofactor. So please pay attention. It can be a cofactor, or this protein part can be a non-protein part can be a coenzyme. What is the difference between a coenzyme and a cofactor? Okay, so cofactor is something whenever you use, okay, whenever you have something like metal ions, okay, not the organic part. Like for example, sometimes you need to have a zinc associated with the enzyme, then only the enzyme is active, right? So when you have, for example, zinc or iron, something like not, it's not an organic part, then we call it a cofactor. Okay, but coenzyme is always and always and always when you have an organic molecule. Coenzyme can never be a metal, it is always an organic molecule. That means it can be any vitamin. Usually we have vitamins which acted as the cofactors. You will come across coenzymes. You will come across with so many vitamins like NAD positive, right? Riboflavin, right? Niacin. They're all used as a coenzymes. So when you have a protein part called an apoenzyme, and when you have a non-protein part, it can be either a cofactor or a coenzyme. It depends upon what enzyme we're talking about. Then only it gives rise to an enzyme, a big enzyme, a complete enzyme, which is called as a hollow enzyme, and that makes your active enzyme. Otherwise, it is not an active enzyme. Did I make it correct now? One is mineral, absolutely Kazi Hakam. One is a mineral, one is a non-organic part, and the second one is an organic part. So coenzyme is always an organic part, and cofactor is a metal ion. But remember, both of them are important for the biological activity of an enzyme. Are we all okay till now? And then I'll tell you one last thing about it. Are we all okay? Did we all understand what is a hollow enzyme? So you can say that cofactor or coenzyme, they are like the helpers, who are helping the apoenzyme. Now you might see that some books they write like hollow enzyme is an apoenzyme plus cofactor. They call coenzyme also a cofactor, but that it depends from one book to the other book. But this is the real definition. Sometimes they say that, oh, cofactor We need an apoenzyme plus cofactor, right? But we actually know that it could be either a cofactor or a coenzyme. Okay, then they say that cofactor can be an organic or it can be an inorganic. Organic like a vitamin, or inorganic like a zinc and iron at all, right? So don't worry if you read somewhere like apoenzyme plus cofactor gives the holoenzyme. That is also partly correct. Are we okay till now? Absolutely. Cofactor and coenzyme have the same function. Your enzyme is not going to be functional unless and until it has a cofactor or a coenzyme. It depends upon, which enzyme you're talking about. It varies from one enzyme to the other enzyme. Okay. So apoenzyme, which is inactive, along, see, cofactor, but you have to know that it could be either a cofactor, or if it's organic it can be a coenzyme as well. So it gives a holoenzyme, which is the active one. Clear? No, it doesn't need both. It might need just the cofactor, or it might need just the coenzyme. Do you understand what I'm trying to say? But yes, one of the two is very, very important for the expression of a particular enzyme, for an enzyme to be active. Okay, but remember, if the question says that which part of the enzyme is a protein part, okay, so it's always an apoenzyme. So enzyme without its non-protein part, which can be either a metal ion or an organic molecule, is termed as an apoenzyme, which is inactive. Remember, apoenzymes are inactive unless and until they bind to a coenzyme or a cofactor. Are we okay? Okay, moving on next, the enzyme activity. Okay, so we will do certain experiments basically, okay, to find out, you know, which what is important for a particular enzyme. Okay, we'll try to know, is it a substrate which is important? Now you know what is a substrate. What is a substrate, guys and girls? How will you define the substrate on which the enzyme acts? That is a substrate, isn't it? Substrate binds to the enzyme, right? Yes. Non-covalently. That binds to the active site. Absolutely. That binds to the enzyme. Absolutely, right? So here we'll do some experiments, kind of, we'll try to find out what is important for the enzyme. Is it enzyme concentration which is important? Is it substrate? Is it temperature, or is pH? So let's change something or the other thing, and then see what's going to happen. Which factors will influence the enzyme activity? Let's try to see that. So in the first thing, what we did is we are trying to see the effect of the enzyme concentration. We're trying to see that, do we really need enzyme for a reaction? Okay, what will happen if you're going to increase the enzyme, the quantity of the enzyme? Do you understand what I'm trying to say? So, but when we use this, okay, what do we do with the others? The substrate concentration, the temperature, the pH, everything is kept constant. So, guys, all these graphs are very important, right? So remember this. This is, these are very important. So you should know all this, okay? So please pay attention, everyone. So here we see that, okay, how important is the enzyme concentration? Okay, what will happen if we double, triple the enzyme concentration? Okay, what we saw that if we keep substrate limited, temperature will keep normal, pH we don't change, substrate we don't change, but we just change the enzyme concentration. I might use first of all 2 millimolar, then 8, then 16, then 30. What's going to happen? We saw when we plotted a graph, we saw that the rate is increasing linearly. The velocity of the reaction is increasing linearly, right? And we saw that the rate of reaction is directly proportional to the concentration of enzyme. The more the enzyme you add, what's going to happen to the reaction? The velocity of the reaction is going to be increased. So concentration of enzyme is very, very important. Did you all understand what is its effect when we keep other things constant? But then let's do the second thing now. Let's keep the enzyme concentration constant now. Okay, let's keep the enzyme concentration constant. Let's increase the temperature and increase the substrate construction concentration, and keep the temperature, pH, enzyme, everything constant. What do you think? What's going to happen? What's going to happen if you increase if you simply increase the substrate construct concentration? Remember, the graph says where initially the rate will increase and then it will be stagnant. Yes, it will reach a point where the rate will stay. Absolutely, it will reach to its saturation point. Very true. you know so what's happening, for example, okay, if I take, for example, I have one test tube, another test tube, another test tube, okay, test tube 1, 2, and 3. What do? I keep the quantity of enzymes same in all the test tubes. Maybe I used 5 molar enzymes here, 5 here, 5 molar enzymes, 5 molar here, right? But then what do? I simply increase the substrate concentration. I might have, for example, I might used here 1 mole of enzyme. I might use here 3 mole of enzyme, or I might use here, for example, 10 mole of enzyme, right? Sorry, yeah, 10 mole of enzyme. Okay, sorry, 10 mole of substrate. Sorry, 10 mole of substrate. So enzyme, the green one is constant. Enzyme, I kept as constant, but here I changed the substrate. I I had less substrate here, but then I had more substrate here, but then I increased even further here. What did I see? I saw that initially I was happy, right? Because my reaction was increasing. But then there was a point when it starts declining now. Or even if it didn't decline, it remained same. Do you know why did it remain same? It remained same because at that time there was no more active sites for the substrates to bind, because enzyme was limited. I just used 555 molar enzyme here, so that means enzyme was limited. There are no binding sites. Absolutely, the enzyme will be used up. So absolutely, because for example, initially I had, for example, one, two, three, four, 5 molecules, okay, of the enzyme. Then I had, every time I had 1, 2, 3, 4, 5. I had 1, 2, 3, 4. I had, every time I had same molecules of enzyme, but what did I change? I changed the concentration. I first used only 1 mole. That means this was bound to this enzyme. Let me use another color. That means it binded to this one. It was bound to this one, right? And second time I increased, I used 2 moles of, substrate. So 2 moles of substrate were bound here. Then I used 5 of them. At this point, all 5 molecules of substrate were bound to the enzyme. Do we have any other empty site for the enzyme? There's no active site for the enzyme. So after this, even if I'm going to make it, like, if it was 5 here, if I'm even going to make it 20, even if I will have 1, 2, 3, 4, 20 substrate molecules, but unfortunately there is no active site because all the active sites are already bound to the substrate. So that is why the reaction is going to stay constant. It's not going to increase further. Okay. Then temperature and pH is very, very easy, because we know that every enzyme has, a constant temperature on which it can work on. Usually the enzymes in our body, they work at almost, 37 degree of temperature, right? This is their optimal temperature. Any temperature beyond that, okay, is going to denature the enzyme, right? If you remember, we saw that we don't need a denatured enzyme. Denatured enzyme will be just a random coil. It is of no use to us because it does not have a secondary, tertiary, quaternary structure. So we don't want to use an enzyme which has lost its activity. So what happened when we kept everything constant, right? But we changed the temperature. When we changed the temperature, we saw that we got a bell-shaped curve wherein initially the activity increased and then it started decreasing. Why? Because now the enzyme was denatured, right? It was deformed. So when it was deformed, substrate was not able to bind to it. Okay, that's why there's something called as an optimum temperature for each and every enzyme. So then certain bacteria, they work at a very high temperature. If you remember, recently I gave you an example of, when we were discussing about Taq polymerase, PCR. If you remember, I told you why we use Taq polymerase in PCR, because that enzyme, lives in hot springs. That enzyme which we got from the bacteria, okay, so that bacteria lives at high temperatures of 100 degrees Celsius. So that is why we took the Taq polymerase to do the PCR, because it can survive at higher temperatures like 100 degrees Celsius. And same as the case with the pH, because we need an optimum pH and optimum temperature, then only the enzyme will be active. That is why any change in our pH in the blood below 7.4 or more than 7.4 is going to be, going to create a drastic change, because the plasma proteins will be denatured at low pH or a very high pH, right? Is it clear to everyone? Except the pepsin, pepsin in our stomach, okay, it works at a low pH. Yes. Oh my god, I was about to say the Muhammad said the same thing. Yeah. Yeah. So the except the pepsin in our stomach, it works at a very low pH, like 1 to 2. Okay. And then we have also one more enzyme in our body, alkaline phosphatase. It has a pH of like a 9 to 10, very very high pH. Okay. And then again, the definitions, wherever you see the definitions, you should know all the definitions. Okay, we're going to talk about these definitions in a while. Okay, so activation, any process that initiates or increases the activity of enzyme is an activation. Inhibition, any process that makes an active enzyme less active or inactive is an inhibition. Then what is a competitive inhibitor and non-competitive inhibitor? Very important. So before we study the competitive inhibitor and non-competitive inhibitor, we need to know something about the what Fisher said and what the Koshland said. Okay, the two important theories. Then only we'll be able to understand these inhibitions. Okay, the competitive and non-competitive inhibition. Okay, so what is a lock and key model? Lock and key model was given by Fisher. Every chapter we are reading about Fisher. Fisher was such an important person for biochemistry, for organic chemistry. Okay, so in the lock and key model, Fisher said that, okay, the enzyme and substrate fit to each other. The enzyme and substrate, they fit to each other in such a way, the way we have a lock and a key. Substrate fits on the enzyme. We have an active site for the substrate where the substrate can bind to the enzyme, but the way they bind, that is like a lock and a key, right? An active site has restricted, only one substance can bind to it. That was said by Fisher in the lock and key model, but later on it was discouraged. It was said that no, it's not like that. The site on the enzyme is not restricted. Okay, why? uh, it was basically later on said that by the induced fit model. Induced fit model was given by a scientist known as a Koshland. Okay, what did he say? He said that any conformational change which takes place in the active site of the enzyme, okay, depends upon the substrate as well. He said that the active site is flexible. The enzyme changes its shape so that the substrate can, absolutely. He said that the active site of the enzyme is not kind of, restricted. He opposed to what was said by the Fisher. Okay, he said that no, this active site is not very restricted. Rather, this active site can change its conformation based upon the substrate. Okay, he gave an example of the glove and the hand. Okay, so he used that, when we put the hand into the glove, right, so the glove is opened further. Okay, the more you put your hand inside the glove keeps on opening, right. Similarly, he said that the conformational change occurs in the enzyme when substrate binds to it, right. So that was about the what was said by the induced fit model given by the Koshland. Is it clear? Did you all understand this? Everybody? We can then go ahead and talk about enzyme inhibition then. Okay, is it okay? Okay, moving on further, then we have the what is the competitive inhibition and non-competitive inhibition. Again, as I said, that every slide of this chapter is so very important. It forms the base of everything, and trust me, this is very easy to memorize. But I have seen that students do get confused when it comes to, okay, what is an active site? What was said by Fisher? What is an induced fit model theory? Okay, what is the basic difference between competitive and non-competitive inhibition? So even though it seems to be very easy, but when the questions are there, they tend to make mistakes. Okay, so please pay attention. So in competitive inhibition, okay, what happens? There's a competition between what? Yes, guys and ladies.
The inhibitor and the substrate.
Yes, so what's going to happen here? There's a competition between these two. Let's see who's going to win. Active site and the substrate, right? So when there's a competition, okay, between the two, okay, for this particular active site between substrate and the inhibitor, okay, so what happens if the competitor, like for example inhibitor, okay, if inhibitor enters the site, let's see, if you see here, this inhibitor has already entered into the site,